Hello everyone. So as we all know that TSPSC JL exams dates are out and Gurukula also going to release the exam dates very soon. So if you consider from the June 1st till September 10th, I have prepared 100 days timetable for complete botany for the JL as well as the DL. If you follow this particular 100 days timetable very strictly, undoubtedly you can crack the exam very shortly. Even though you did not prepare so far, even though you start your preparation from the June 1st, if you follow this 100 days, very easily you can crack the exam. In this particular video, I will help you how to prepare each and every day. I will provide you 100 days timetable day wise. At the same time, I will provide you complete information about all the 10 units in the botany and what kind of books to be followed and what is the method of reading every specific chapter. So I'm requesting every student, please watch the video till the end. You will get a wonderful information for your preparation. So the new batch is going to start from the June 1st onwards for the botany as well as the zoology. If you are truly passionate about cracking this particular JL or DL, please enroll in this particular batch and you will get 1 week of completely free classes. You will get all the live and recorded classes, complete coverage of 10 units, and you will get a 10 chapter wise test and 10 mock test. I will give you the schedule in this particular PDF only, and you will get more than 2500 practice questions also. Okay, so now first, I know whomever writing this particular JL and DL exam, most of them are working teachers. Every one of you know this, right? So to help you in effective preparation, I have segregated entire 10 units of syllabus into 100 days along with the chapter wise test, along with the mock tests also, right? So suppose example, so how we are covering for a big chapters, we are keeping 10 to 12 days. For small chapters, we are keeping 7 to 8 days. So what I have done, I have segregated all the 10 units into 90 days, and remaining 10 days we have allotted for the 10 mock tests as well as the doubt solving sessions also. So how I have made the timetable, because the disadvantage with some teachers and some students is that how they will read is they will keep 10 days for one chapter. See friends, you should not read in this particular way. What is the reason? If you are focusing 10 days only on one chapter, one first thing is that you get bored because you're reading the same chapter. You don't get much interest, and most importantly you are unable to cover vast amount of the syllabus in the less period of time. So that is the major reason what I have done. See here, in a single day we are covering three different units. Suppose example, the batch is starting from the June 1st. See here, we have covered Unit 9, plant physiology, Unit 7, cell biology, Unit 4, plant anatomy and embryology, right? So in this way, we are segregated the entire syllabus. So see here, so in this manner, by the 15th of this particular what we can say 15th June, we are going to complete entire plant physiology, cell biology as well as the anatomy also. So in the 15 days we are covering three chapters very easily. And how many hours you have to spend? You have to spend hardly 3 hours of classes, and minimum at least 2 to 2.5 hours on preparation. I am telling you, every student or every teacher who is preparing for the exam, I know many teachers are having hectic schedule and many people are housewives also. But if you want to crack this exam, these 100 days, you have to spend minimum 6 to 8 hours. But if you're a working professional, at least you have to spend 4 to 5 hours, which is mandatory, compulsory. You have to spend it. Otherwise, you never crack the exam. I'm challenging you. See here, revision multiple times we have to revise the concept. By that you can easily remember. So after 15 days we are finishing the three units. Immediately we are going to write the particular test on that particular three chapters. After 15 days, once again I am covering the 2 to 3 big units. Say an example, after that we are covering the very big unit ecology and the unit 1 phycology, mycology, bacteria and viruses. So see here you have to spend only 2 hours on classes, and remaining 3 to 4 hours you can do the vigorous revision, right? So after that, see how I have prepared the timetable is one easy chapter, one difficult chapter, otherwise one difficult chapter, one medium level chapter, because you people has to focus up to the MSc level. So that is the reason you have to read very depth of the concepts also here. So see here, I have covered rDNA technology with bryophyta, pteridophyta and gymnosperm. Bryophyta, pteridophyta, gymnosperms, you have to remember abundant amount of examples, their reproductive methods, their habitat, their economic importance, everything, their life cycle. We have to remember a lot of information. When it comes to the rDNA technology, it is purely conceptual based. So that's why I have designed it in such a way that in one way you can read pure conceptual topics, in other way you can read a pure theoretical, or where you have to remember more amount of the concepts. After the rDNA technology, we are covering genetic engineering. Then in the that is unit 10, I'm going to explain you. We have to cover rDNA technology, plant tissue culture, somatic hybridization, organogenesis, protoplast fusion, right, and plant tissue culture applications, then as well as transgenic plants. So we are going to cover each and everything. In the upcoming, I will explain you what kind of books to be followed. Then after that, see here, I have made unit 5 and unit 3. So this is little difficult. It's why it is difficult. Angiosperms also you have to remember a lot of information, and unit 5 plant resource utilization also you have to remember a lot of information. Among these three, among these two, which is the easiest unit? Undoubtedly, friends, unit 5 is the easiest one. Why? Because here you have to remember in situ conservation, ex situ conservation, like national parks, sanctuaries, botanical garden, biosphere reserves, tiger zones, and you have to know the generalized information about the different national institutes like CSIR, NBIR, NBRI, all this, which is a very easy. And only you have to focus on the some medicinal plants and some theoretical concepts such as the ethnobotany, right? So in this particular period, you have to focus more on the taxonomy of angiosperms. After that, once again, we kept molecular biology with angiosperms. Then after that, I kept genetics. Why I kept genetics and biochemistry as a specialized chapter? Because cytogenetics is a little difficult concept. So that's why we have to read, alone, and we have to spend more amount of time actually. Okay. So after completion of all the chapters, now you only see here, suppose example, every student has to watch this compulsory. Test 1. Suppose we finished cell biology, plant anatomy and embryology. Immediately after completion of these 3 chapters, you will get 1 day of time. You have to write the test. Once we finish plant physiology and ecology, you have to write a test. So you will get all the chapter wise test. Then friends, apart from the chapter wise test, we are covering all the 10 units of botany from very briefly to advanced level. You have to read it compulsory. Then we are writing 10 complete mock tests also. After this, even I will provide you the complete doubt solving sessions also. Apart from chapter wise test, apart from the mock test, I will help you in providing the 2500 MCQs. So we are having 10 units in botany, is it? So out of this particular 10 units, for each unit, I will provide you the 250 MCQs, right? Hence, how many MCQs you can answer? 2500 you can practice. And we are having 10 chapter wise test. There you will get the 500 questions. And if you look into the mock test, you will get the 100 questions from the mock test. How many mock tests are there? Total 10 are there. So total, before 100 days, you are going to complete 4000 MCQs. If you really follow this 4000 MCQs thoroughly, trust me, you can crack the exam very easily. And why most of the students never crack the exam? What is the reason? There are only 2 reasons. What is that particular reason? They never ever understand how to study. First point. Second point, they never ever analyze the syllabus. I am not joking, friends. If you do not analyze the syllabus very correctly, you never ever crack the exam. 90% of the success rate depends on analysis. If you analyze it thoroughly, undoubtedly you can easily crack the question. How to analyze? You have to analyze the syllabus. How? With the help of PYQs, at the same time with the help of applicative knowledge on that concept. Because last 2014-15, that time your syllabus is not PG level. So that time the syllabus is UG level. Now this time the syllabus is PG level, because you are the one who is going to write this first time. So you have to read compulsory very depth. Then only you see. If you read depth, if the questions are from basic, there is no problem. But if the questions are from depth, it will be great advantages to you. So that's the reason when you are reading every single chapter, read from very basic to very advanced. Now I am going to explain to everyone how to study every single chapter. Okay, every one of you, please follow me. Okay, listen. So now let's take plant physiology and biochemistry, which is one of the biggest unit in the botany. You can expect 10 to 15 questions easily. So now most of the students will ask me, sir, I have only Telugu Academy book. Is it sufficient? I can tell you, friends, it is not at all sufficient, because you have to read very depth. Right. If you look into the biochemistry in the Telugu Academy, it is just written what is glycolysis, what is the steps involved in the glycolysis, what are the enzymes. What about the regulation part? How the Krebs cycle get regulated? Which inhibitors can block the electron transport chain? That entire information was not given. So you can read either Pathfinder or Plant Physiology. The standard reference book is the Lincoln Tace. If you are not finding a time to read the Lincoln Tace, strictly follow my PPT. First point, follow PPT and read the PDF whichever I already provided to all of you. The third and most important, you have to practice MCQ. There are two type of students are there. One type of student, they will only read the content, they never practice the question. Second type of students are there, they're only focus on MCQs, they never know the concept, they never study also. These two type of students will fail in the exam. If you want to crack the exam, first thing, you require wonderful knowledge on the concept. Second thing, practicing MCQs. When you have a knowledge, when you practice MCQs, undoubtedly you can crack the exam. There is no doubt in that. So how to study this particular plant physiology? So let's start with the photosynthesis. When you are reading the photosynthesis, first you have to read all the type of pigments. What are antenna pigment or accessory pigment? What are reaction center? What are chlorophyll? Their types, their structures, xanthophyll structure, carotenoid structure, where they are present. Most important it is. And what is the difference between the cyclic photophosphorylation, non-cyclic photophosphorylation, where they will occur, how many number of ATP we are going to produce, where the light reaction occur, where the dark reaction occur, what is the fundamental difference between the C3, C4 and CAM plants. In the C3 plant, how many cells are involved? In the C4 plants, how many cells are involved? CAM plants, how many cells are involved? Which plants are avoiding the photorespiration? How many number of ATPs are produced? Everything we have to read. So for that reason, in order to study this much depth, you have to study from very basic to very advanced level. Then obviously you can crack the question. So you have to understand 10 to 15 question means such a higher weightage it is. So you should not miss even a small topic also in this particular plant physiology and biochemistry. Solute transport, very common topic. You have to focus on structure of a xylem. What does the xylem made up of? Xylem parenchyma, xylem tracheal elements consist of a tracheids, vessel elements. Phloem consists of phloem parenchyma, sieve tube elements, sieve cells. What are companion cells? Where they are present? What are the P proteins? How does the cohesion tension theory will work? You have to read compulsory. How does the Munch flow hypothesis will work? Whether if the sugars want to be transported, will they use any kind of a active transport or passive transport? And how does the sugar can be loaded? How does the sugar can be unloaded? How the water molecules are moving? Everything you have to study from very basic to very depth. And the most important among all, plant hormones, where you can expect minimum 2 questions, because it's a very broad concept. Whenever you are reading, majorly focus on auxin, cytokinin, gibberellin, abscisic acid, ethylene. Apart from that, give a little importance about the jasmonic acid, salicylic acid, and polyamines as well. So these 8 hormones you have to focus, but first 5 hormones are utmost important. Okay, after this, how to study the hormone? First, name of the hormone, which biosynthetic precursor is involved in the synthesis of that particular hormone, biosynthetic pathway, transport, precursors involved over there, functions, applications. These are 7 things you have to study for every single hormone. Beyond that, there is no way of asking the question. Nitrogen metabolism. Mostly in the nitrogen metabolism, they can ask the question from either from bacteria or from the processes or from the genes or from the enzymes. Only 4 ways are there: bacteria, genes, enzymes, and processes. What is nitrogen fixation, nitrification, denitrification, ammonification, assimilation, where they will occur, which amino acid is involved, what cofactor is required for nitrogenase enzyme, what cofactor is required for the nitrate reductase enzyme. You have to read all these along with the bacteria, which are example of symbiotic bacteria, which are the free living bacteria, which are anaerobic, which are aerobic, which are obligate anaerobe, which are obligate aerobes, facultative aerobes, facultative anaerobes. You have to read along with the multiple examples. Then secondary metabolites. You have to focus on the secondary metabolites, majorly remembering the information. What are phenols, what are terpenes, what are nitrogen containing compound, along with multiple example, because very straightforward question you will get it. Sensory photobiology and stress physiology. These sensory photobiology, pure conceptual. You have to study about phytochrome, cryptochrome, phototropins, their structure, their function, and genes. Stress physiology, friends, you have to, it's a kind of a dry topic, kind of theoretical. You have to majorly focus on oxygen stress, salinity, chilling, and osmotic stress. These 4 stress are extremely important. So these are the topics you have to study in the plant physiology. When it comes to the biochemistry, see friends, in the botany syllabus, biochemistry is a very small part, but you can expect, I told you, right, including plant physiology and biochemistry, you can get 15 questions, which means that 3 to 4 questions you can expect from the biochemistry. In that, undoubtedly, without fail, you are going to get the question from the enzyme and its kinetics, compulsory, mandatorily. So most of the students will know the classification of enzymes. Then what is active site, lock and key model, inducer fit model, what are the factors affecting the enzyme activity? You all read it, but I want you to majorly give a highest importance to the kinetics. Michael-Menten kinetics, how the kinetics, I mean to say, they can ask the numerical question by giving the formula of the Michael-Menten equation. You read only Michael-Menten, you no need to read LB plot, Eddy-Hofstee diagram. These are all not required. I will explain you how to study while I'm explaining you. See, you can't read Leninger, White and White. They are very big books. You people don't have that much of time. So you people can focus on the Pathfinder, and most importantly, follow my PPT. Very importantly, follow the lesson what I'm teaching. So glycolysis regulation, Krebs cycle regulation, ETC, followed by inhibitors blocking the ETC. Most importantly, production of ATP, NADH, FADH2, GTP. I want you to remember even carbon dioxide also. How many carbon dioxide are releasing, how many water molecules are releasing. Not to get confused and not to forget. And when it comes to the lipids, you have to focus on the metabolism also, that is beta oxidation also. You have to remember the chemistry also. Then cell biology. See friends, cell biology, I never say that it is very difficult. See, when compared to the plant physiology and biochemistry, friends, I can tell you that the cell biology is easy chapter only. It's not too difficult compared to that. So you can expect 8 to 10 questions. If anyone wants to read the standard reference book, most of the teachers know. Whomever already working as a DL, so everyone know about this book, Carp Lodish. If you can't read this, follow my PPT strictly. Follow the UG level material, whatever I am giving you, along with the Pathfinder. Enough. You can answer any kind of a question very easily. When you are reading all the organelles, remember, first structure, functions, third one, enzymes involved in the organelle. Very importantly, the deficiency diseases caused by the deficiency of enzymes in the organelle. Majorly focus on plasma membrane, RER, SER, nucleus, mitochondria, peroxisome, Golgi complex, utmost important. Then followed by the cytoskeleton, lysosomes. These 9 organelles are extremely important. You have to read structure, function, enzymes, and their deficiency diseases. Apart from this, you have cell division also. Undoubtedly, you are going to get the question from cell division. Undoubtedly. Along with the cell division, my friends, it is very important you have to focus on the cell cycle regulation. How the cell is getting regulated, the G1 phase, S phase, G2 phase, M phase. what are the CDKs, what are the cyclins involved in the different phases of the cell cycle, all you have to read it, compulsory, mandatorily. And you should also focus on a specialized concept such as transposon, structure of euchromatin, heterochromatin, telomere, centromere. All these you have to read compulsory. Very easy chapter, scoring chapter, but the tricky questions can appear. Then cytogenetics. I told you, right? 90% of the students will not answer the questions from this topic because of the difficulty level. That's why if you answer the questions from this topic, you can easily score better when compared to other. So you have to read Pathfinder, Watson, PJ Russell, all this. But you can't get this much of time to read all this. So you can focus on the Pathfinder. Very important. You can focus on the Pathfinder, then followed by my PPT, and most importantly, lesson. PPT is secondary. First, you have to learn the concept. Whatever I'm speaking in the classes, that is more important from where you can expect the questions. Right. Then, friends, how to read. When, suppose example, if you're reading the replication, first remember, follow the rules, followed by the enzymes, followed by the process. Always first read prokaryotes. Directly don't go to the eukaryotes. Because if you understand the prokaryotes, eukaryotes become easy. Then followed by transcription. Same with prokaryotes and eukaryotes. Very important concept is the post-transcriptional modifications. You have to focus very depth on the 5' capping, 3' polyadenylation, then followed by the splicing. Apart from that, it is very, very, very important is translation. Protein synthesis in prokaryotes, eukaryotes. Most of the students will leave the post-translational modifications. Along with the post-transcriptional, you have to read the post-translational modifications also. And give highest importance to all the enzymes involved in replication, transcription, translation, their structure, their subunit, everything you have to read. Most importantly, I'm requesting every student to focus on the antibiotics. Remember my words, antibiotic blocking the translation, transcription, replication. You have to remember them. Right. And followed by DNA repair mechanisms. That is also extremely important. Friends, cytogenetics, including molecular biology and genetics, you can expect easily 10 to 15 questions. So it's a very important unit. And we are about to finish the genetics also, right? You first start with structural and numerical alterations. Read all the deletion, duplication, inversion, translocation. Focus on example. Focus on types. Types is very important. Then, friends, followed by the all ploidies, polyploidy, aneuploidy, nullisomy, trisomy, monosomy, tetrasomy. Most importantly, you have to remember the example. Then you focus on the cistrons test, complementation, what is incomplete dominance, codominance, multiple allelism, pleiotropy, sex limited, sex influenced characteristics you have to study compulsory. And botany people, I want you to also focus on Mendel principle, especially focus on monohybrid, dihybrid, trihybrid. It's not mandatory. It is not mandatory because it is not mentioned in our syllabus, but you have to read compulsory mono, di, trihybrid. It's mandatory. Now next, ecology, one of the biggest unit. You can get maximum number of questions here also. And you know very well, studying ecology is not a small thing. It's a very big unit, right? So we have to focus from I never encourage any student to leave anything in plant physiology and biochemistry, and I don't encourage any student to leave anything in ecology. It's a mandatory unit. Start from individual. So read the habitat and niche, read complete population ecology, read complete community interaction, starting from predation, competition, amensalism, commensalism, mutualism, everything I want you to study compulsory. Then Lotka-Volterra model, then followed by the ecosystem ecology, where you are going to read food chain, food web, pyramids, and different type of ecosystem like terrestrial, aquatic, grassland, their characteristic feature, what is the rainfall, everything you have to study. Very important and unique concept, ecological succession. What is primary, secondary, heterotrophic, autotrophic? What is the principle of succession? What is the mechanism of succession? What is the succession that is occurring in the bare rock? What is the succession occurring in the saline water? Everything you have to read. Followed by the pollution, especially air pollution, water pollution. In the air pollution, majorly remember the gases. In the water pollution, majorly remember the BOD. Not to get confused with that. Then followed by conservation biology, where you can expect 2 to 3 questions, which is linked with unit 5 also. In situ, ex situ. You have to read national parks, sanctuaries, biosphere reserve, tiger zones, which are critically endangered, vulnerable, near threatened, least concern, red data book, all the information you have to read from very basic to the very advanced. I know if you want to read this, you have to read entire ecology by Verma and Agarwal and Odom. If you don't find the time, read the PPT, follow the Pathfinder. More than sufficient. Then plant biotechnology and genetic engineering. Here also you can expect 10 to 11 question. Very important unit. See, being a botany student, you have to read 10 units. There is no doubt about it. But I am explaining you, with respect to the weightage. T.A. Brown is the standard reference book for the plant biotechnology and genetic engineering. But you don't find the time, you follow my PPT strictly and follow the PDF that what I'm providing you. Recombinant DNA technology. You have to focus on what? All the enzymes starting from, starting from what? The enzymes which will cut the cell wall, like suppose a chitinase enzyme, as well as cellulase enzyme, as well as the enzymes which are going to cut the DNA. I mean to say exonuclease enzyme, endonuclease enzyme, type of restriction endonuclease, type 1, type 2, type 3, polynucleotide kinase, terminal nucleotide transferase enzyme, which we can call modifying enzymes. You have to read compulsory, followed by all the vector, plasmid, casmid, bacteriophage, bac, yeast artificial chromosome, everything. MAC, mammalian artificial chromosome, all you have to study along with their characteristics, followed by most important is application. Then genomic and cDNA library. Then friends, what kind of questions you can expect in the plant, cell and tissue culture? Mostly statement related question or straightforward questions only. I mean to say, you have to study the type of tissue culture. What is the hormone composition that we used in the tissue culture? What is the artificial seed formation method? How can we produce the soma clones, and what is their application? How you can produce the natural products or secondary metabolites? What is the methodology of the germplasm storage? Apart from this, I want you to compulsory follow the transgenic plants and gene transfer, micro injection, electroporation method, electric shock method, gene gun, everything. In this, focus on the technique of only three: PCR, southern blotting, and western blotting. Only focus on these three technique. You no need to focus on any other technique. And plant resource utilization and conservation. Remember, every student, please look into this. From here, you people are going to get compulsory 2 questions. Guarantee. From these institutes only. BSI, NBPGR, ICAR, CSIR, DBT. I will tell you. What you have to remember, I will explain you in the lesson. When they established, which year they established, what are their scopes and objectives, means what are the functionalities or objective of that particular organization, which year they established, and what are the recent activities that they are conducting. That also you have to remember, compulsory, mandatory, and it is easy scoring. Sanctuaries, national park, biosphere reserve, wetland, mangroves. These are the concepts where you have to use your by-hearting skills, because nothing concept over there. You have to remember what are the biosphere reserves. There are 18 biosphere reserves. In India, there are 9 biosphere reserves are there. You have to remember which state they are located, what is the flora and fauna over there, what is the dominant animal protected over there. Everything you have to remember. What are the top 10 national park? What is the animal protected over there? What is the conservation status? You have to read compulsory. Role of plants in medicine, compulsory you have to remember. Actually, they have given only 4. We will read 4. Compulsory, we will read the 4. And ethnobotany, which is a theoretical concept, we can expect 1 question. I will explain the ethnobotany compulsory because it's a pure theory. It's studying about the scope and advantages. And little bit we have to study about the tannins, dyes, gums, resins, their uses, medicinal value, economic importance, application, raw materials used for them. We have to read all this. Very important unit this is also, but kind of UZ unit. So phycology, mycology, bacteria and viruses. I will tell you one thing, friends. See, if you take an example of IIT JAM, which is an MSc level entrance exam. Okay, if you take a set exam, which is a PhD entrance exam, which is a net exam. In these particular units, even these exams also, you get a question from the examples only. 99% questions from the examples. So Here, I encourage all of you, you can focus on the NCRT, you can focus on Telugu Academy books also. That is not at all a problem because it's a basic level only. Let's consider virus. Remember everyone, whenever you are reading a viruses, first point you have to read characteristic of virus, structure of virus, types of viruses. Most important for the exam, the diseases caused, because in the matching questions we can expect that, you know, right? Virus attack on the plant also, virus attack on the animal also, virus attack on the bacteria also. So you have to remember different type of diseases caused by the viruses with respect to the different organisms. You have to remember compulsory. Let's talk about the bacteria. When you are talking about bacteria, their structure, microscopic structure is very important. What does the cell wall is made up of? What enzymes are present in the cell wall composition, what does the cell membrane is made up of, what is the genetic material, what is the function of the plasmid, what are the type of the ribosomes, what are the function of the gas vacuoles, what are the functions of the mesosome, what are the functions of the pili and fimbriae, what are the functions of the flagella, what are their nutritional types, what is the reproductive methods. Everything A to Z you have to study compulsory. When it comes to the mycology, same. Remember first thing, first for anything, whether it's a bacteria, fungi, protista, or bacteria or virus, all the time remember first to focus on the structure, then followed by the ultrastructure, means microscopic structure, their types, then followed by nutritional parameter, whether it's a autotrophic, symbiotic, biotropic, saprobic, and what is the type of reproductive method. Is it following vegetative, asexual, sexual? If it is asexual, what is the type? If it is the sexual, what is the type? If it is a vegetative, what are the types? Most important, economic importance. Economic importance and environmental importance also extremely important for the exam. Right. And in the algae also majorly focus on the Phaeophyta, Chlorophyta, Rhodophyta, Xanthophyta. These four are more important. And give a highest importance in remembering toxic algae. Examples of toxic algae, what kind of toxicity that they will cause. That also you have to remember. I don't encourage anyone to leave any topic from this also, because easy topic, scoring topic. Okay. Now, some students are also having the doubt on the how to study the anatomy, how to study the embryology, how to study the gymnosperms and angiosperms. Okay, I think I forgot to keep that particular slide. Okay, now listen. When I will explain in the class also, when you are reading, let me open you the syllabus copy, by that you will get easy idea. See, where you can expect more number of questions. Will you expect the more number of questions from the gymnosperms, or will you expect the more number of questions from the angiosperm? I can tell you friends, more number of questions will come from the angiosperms only when compared to the gymnosperms. Okay, it doesn't mean that you have to neglect this particular gymnosperm. No, you have to give the highest importance, but give top priority to the angiosperms only. And remember, every time you can expect at least 1 to 2 questions from the species, genus, family, order. Right? See, because they will give four to five examples in the matching column, and they will ask us, please arrange them along, with respect to the taxonomic hierarchy, which comes in the species category, which comes in the genus category, which comes in the family category, which comes in the order category. So that's how you have to remember them compulsory, because at least one question you can expect from here. And trust me, ICBN is also very important, friends. Remember what is international code for botanical nomenclature, which year it was introduced, what it is explaining us. Based on the ICBN, what is the classification of the angiosperms? You have to read compulsory. Compulsory ICBN. In our competitive exam, remembering the organizations are very important, friends. At least you can expect 1 to 2 questions compulsory from the organization. And taxonomic tools, I am requesting you majorly focus on the herbarium. Majorly focus on the herbarium, histological and cytological. These three are most common, widely asked. If you can leave the phytochemical, serological, biochemical also, but majorly focus on the herbarium, histological and cytological tools. Compulsory you have to read. Then angiosperms. I told you, right? Whenever you are reading the classification, the most important point you people has to remember for the exam is examples, right? So how many number of classifications are there doesn't matter. And how to remember the classification, most of the students try to by heart the classification. Don't do that. I am requesting to make a chart. If suppose example, there is a Magnoliaceae family, Malvaceae, Rutaceae, Apocynaceae, SLPDaceae, Lamiaceae. There are examples, 7 families are there. So friends, you make one chart for the 7 families. For 7 families, make only one chart. What is the floral formula? What is the estivation? Most importantly, focus on the diagrams, because the question will ask from the diagrams only. What kind of androecium, what kind of gynoecium? Most important floral formulas you remember. In the exam, directly they will give the floral formula and they will ask you this floral formula belongs to which family. And if you read, you will forget. So that's why take one chart or a paper and write down 7 families, different characteristics, and write down different characteristics for the 7 families. By that you can easily remember it. So the best way to remember the families is making the chart. Don't read and waste the time. Make the chart and try to visualize it every day. Then plant anatomy and embryology. When you are starting here, I am talking when you are doing the preparation, start from the pollination. Pollen pistil interaction. 90% of the time they will raise the question from the pollination mechanism. Most importantly, vectors used. They can ask which of the following vector used in this specific pollination mechanism. And remember what is the double fertilization also. Then after the pollination, you directly come to shoot development, root development. Right. In the shoot development, majorly focus on the organization of shoot apical meristem, the different parts of the shoot, how they are organized. Different parts of the root, how they are organized. Suppose if it's the root, how does the xylem got differentiated? How does the phloem got differentiated? When the xylem will get differentiated? What is the endarch xylem? What is the exarch xylem? Where the endarch xylem is present, where the exarch xylem is present. How can you identify the endarch xylem? How can you identify the exarch xylem? You have to read compulsory. Then followed by, this is a kind of new topic for you, cell to cell communication. I will teach this little depth. You have to read this. Then for same with the root also. So first to focus on pollination, then read the shoot and root development. See, phyllotaxy is all about examples. It's your wish. If you want to remember, remember. Otherwise, you can neglect. It's not greatly important when compared to other. Then I never encourage anyone to leave these 3. Male gametophyte structure, female gametophyte structure, seed development, and fruit growth. If you are, whenever you are reading the male gametophyte, remember my words. You have to remember the question with respect to the structure. What is the exact role of the tapetum? What is the how does the pollen development occur? Remember everyone, in the pollen development, what kind of genes will be expressed? That is important, friends. What exactly male sterility means? When we can call a condition is a male sterility. How does the pollen tube will grow? What is called pollen allergy? What are the different type of pollen embryos? I don't encourage any one of you to leave anything in the male and female gametophyte. Leave everything. Sorry, read everything. Every single point you read it. And plant anatomy, embryology also you can read the Telugu Academy level. If you find it is not sufficient, you can ask me. I will provide you the PDF of the PG level. But trust me, in case of plant anatomy and embryology, you never ever get any application based or a logical indirect question. You will get a very direct question only, friends, but you will get the question from very depth of the concept. So if you want to answer that kind of a question, you have to know, suppose example, a question asked from the pollen germination. If you want to understand the pollen germination, first of all, you have to understand the anther. What is the structure of anther? How does the pollen is getting developed? Then when you understand how the pollen is getting developed, then only you will understand the pollen germination. So that's why it is not ideal as you to leave anything, read everything in the male gametophyte, female gametophyte, seed development. In the seed development, majorly focus on the storage protein, because you can expect the matching questions from the storage proteins as well. So don't skip a male gametophyte, female gametophyte, seed development, never ever. Next, gymnosperms. What I told you, friends, first give the importance to the angiosperms, then come to the bryophyta, pteridophyta, gymnosperm. Same thing I am saying. There is no very depth conceptual question. You will get the conceptual question, a straightforward only. I cannot say that it's a truly logical, a little bit logical. First thing, whether it's a bryophyta, pteridophyta, gymnosperm, first highest priority will be the classification. First one, followed by structure, followed by reproductive method, followed by economic importance, followed by ecological importance. Once again, remember, structure, reproductive methods, classification, economic importance, ecological importance. Here also, friends, I don't encourage you to read for multiple times. Instead of reading multiple times, make a chart. If you make chart one time, you can visualize it 100 times. Easily it can be imprinted in your brain. Always try to remember these information in the form of a pictures rather than remembering in the form of a words. Okay. Okay. So that is all the important topics that you have to study. And I'm requesting every aspirant, be sincere with this 100 days. Undoubtedly, we are going to crack the exam. Okay. Just be sincere with your preparation for 100 days. If you are not working, you have to spend 6 to 8 hours compulsory. If you are working professional, you have to spend minimum 4 to 5 hours. Then only we can crack the exam. Okay. So thank you all. I hope the discussion is really useful to all of you.